Recall that in the last lecture, we discussed the specification of the different germ layers in the amphibian embryo, and we began discussing the specification of the different axes of the body. And we saw how experiments that were conducted by Hans Spemann and Hildy Mangold show that a region of the amphibian embryo, the dorsal lip of the blastopore, and the structures that derive from that region make up what is known as the amphibian organizer. And this tissue was given the name organizer because it has the ability to induce the formation of a secondary axis when it is transplanted to a second embryo. In this lecture, we'll explore the mechanisms by which the organizer forms and acquires its properties, as well as the mechanisms by which it induces the formation of the neural tube and the specification of the various axes of the body. Experiments in which different portions of the blastula were recombined show that the organizer is induced by signals that come from the underlying endoderm. As we've seen, the organizer forms from cells that are located at the marginal zone or equator, which is where the animal and the vegetal poles meet. If we take an animal cap, which is represented here by the letter 1, and normally gives rise to the ectoderm, and we separate that from the embryo, mesodermal structures do not form. Similarly, when the vegetal pole cells are separated from the embryo, mesodermal structures also fail to form. However, if we take an animal cap and combine that with a portion of the vegetal hemisphere, we see the formation of mesodermal tissues. This shows that the endoderm is responsible for inducing the cells of the mesoderm, including the organizer. Another finding of these types of experiments is that the type of mesoderm that's induced by the vegetal pole cells, which are the cells of the presumptive endoderm shown here in yellow, depends on the region of the embryo from which the vegetal cells are isolated. So vegetal cells from the dorsal side of the embryo induce the formation of dorsal mesodermal structures, which include the organizer. On the other hand, ventral and lateral vegetal cells, those that are closer to the site of sperm entry, induce ventral and intermediate mesoderm, things like mesenchyme, blood, and kidney cells. And you can see a graphical representation of these results in this figure. If we combine an animal cap, which is a presumptive ectoderm, with dorsal vegetal cells, the animal cap cells are induced to form muscle and notochord, which are dorsal mesoderm structures. On the other hand, if we combine animal cap cells with ventral vegetal cells, blood cells form instead. So these dorsalmost vegetal cells of the blastula, which are capable of inducing the organizer, have been called the Nieuwkoop center, and that name was given after the scientists who carried out some of these experiments. Further evidence for the function of the Nieuwkoop center came from transplantation experiments in African clawed frogs. In these experiments, scientists took the dorsalmost vegetal blastomeres from one blastula and transplanted those into the ventral vegetal side of a second blastula. When they did this, the resulting larvae had two embryonic axes, so they had two heads. Similarly, injecting cortical cytoplasm from the dorsal vegetal cells of one frog embryo into the ventral vegetal cells of a second frog embryo also led to the formation of a second axis. These results show that the cells that are localized at the vegetal pole and which are at the most dorsal region of the embryo where the back will form are the ones that induce the development of the dorsal mesoderm or organizer. Once scientists determined that the dorsalmost vegetal cells, in other words the Nieuwkoop center, could induce animal pole cells to become dorsal mesoderm and as a result create a secondary axis, they set out to determine which factor gave the Nieuwkoop center the ability to induce the dorsal mesoderm fate. Beta-catenin was found to be that factor. And recall that beta-catenin is a transcription factor that becomes stabilized by the binding of Wnt to their receptors. It was found that depleting beta-catenin leads to the loss of dorsal structures, while the addition of beta-catenin, which is shown here, into the ventral side of the embryo could induce the formation of a secondary axis. So how does beta-catenin end up being present in the cells of the Nieuwkoop center? Okay, so this is where the site of sperm entry ties into the specification of the dorsal-ventral axis. Because the localization of beta-catenin is indirectly driven by the cortical rotation, which is induced by the entry of the sperm. In this figure, green represents signaling proteins, paracrine factors of the Wnt signaling pathway. and the red represents the stabilization of or the activation of the Wnt signaling pathway, which means the stabilization of beta-catenin. As a result of the cytoskeletal rearrangements that happen during fertilization and early cleavage, beta-catenin starts accumulating in the dorsal region of the embryo. It's labeled in orange here in part A. Beta-catenin continues to accumulate in the nuclei of the cells at the dorsal side, which are labeled here in part B, but it's not present in the ventral nuclei. The beta-catenin accumulation in the nuclei of the dorsal cells occurs throughout early cleavage, such that beta-catenin can be found at the Nieuwkoop center and at the organizer regions. So let's look at how beta-catenin gets into the nuclei of the dorsal vegetal cells and how this relates to the site of sperm entry. Proteins of the Wnt pathway, which act upstream of beta-catenin, are initially localized at the vegetal pole of the egg. These proteins get moved to the dorsal side of the embryo during fertilization, and that happens as a result of the cortical rotation. The presence of Wnt proteins in the dorsal side leads to the stabilization of beta-catenin because these proteins act to prevent the degradation of beta-catenin. So what would happen if we were to allow beta-catenin to become stabilized not only on the dorsal side, but also on the ventral side of the embryo? Well, if we were to inactivate a protein that inhibits beta-catenin, GSK3, so that beta-catenin became stabilized in both the dorsal and the ventral side of the embryo, a second dorsal axis would form.